This video covers Advanced Higher Biology Unit 1 Cells and Proteins, specifically Key Area 1 Laboratory Techniques for Biologists. We'll look today at Section C of that key area, Separation Techniques. So describing how a centrifuge can be used to separate substances by density, how thin and paper chromatography are used to separate amino acids and sugars based on solubility. We'll explain the use of affinity chromatography to separate proteins, briefly explain how gel electrophoresis is used in separating proteins and nucleic acids, and describe how native and STS page electrophoresis differs. So firstly, we look at centrifugation. It separates substances based on their density. More dense components will settle in the pellet, while less dense components will remain in the liquid section known as the supernatant. Centrifuges are very useful when we're trying to separate solid components from a solution which they are suspended in. For example, blood cells, like you can see here, being separated from within the blood plasma. Thin and paper chromatography can be used for separating different substances such as amino acids and sugars. In all forms of chromatography, we have a stationary phase, which is the material which stays still and allows the other motile phase to pass through it and draw the separating chemicals along. The speed with which the solute travels along the chromatogram will depend on its differing solubility in the solvents used. There are many different solvents we can use in both the paper and thin layer chromatography, also known as TLC. In paper chromatography, the stationary phase is a piece of absorbent paper, and the motile phase is a solvent. It's very useful to use paper chromatography in separating color pigments, for example, in plant science. Thin layer chromatography is performed on a sheet of glass, plastic, or aluminium foil, which is coated with a thin layer of absorbent material, usually a silica gel, aluminium oxide, or a cellulose. This layer of absorbent is our stationary phase. After the sample has been applied to the plate, the motile solvent phase can be drawn up through the plate along capillary action. Because different analytes ascend the TLC plate at different rate, separation is achieved. After the experiment, the spots are visualized using either UV light or a chemical method. During the run, the mobile solvent will move up the plate quicker than the dissolved sample. So to quantify the results, the distance traveled by the substance being considered is divided by the total distance traveled by the mobile phase, the solvent. This ratio is called the RF value. In general, a substance whose structure resembles the stationary phase more closely will have a low RF value, so will move further in the given time, while one that has a similar structure to the mobile phase will have a higher RF value. Thin layer chromatography can be used to monitor the progression of a reaction, identify compounds present in the mixture, and determine the purity of a substance. So we can see here the solvent front and the mobile phase has moved 2.8 centimeters and the new position of the compound is 2.1. So you would find your RF value with 2.1 over 2.8, which is. Process of affinity chromatography differs from paper and thin layer chromatography. In this, we rely on the binding interactions between a protein and specific molecules bound to a matrix or gel. A specific molecule is immobilized on an inert support in a column, and protein mixtures pass through that column. The target protein, which is complementary to the specific molecule in the column, will bind to it and remain in the column after the other components have washed through. So you can see here the hexagons are able to stay within the column because they're bound to the receptor, and all the other ones, your squares and circles and triangles, they go right through. So we can elute out, the verb, all the ones that won't stick, and then separately we can now release the desired proteins and they've been separated or purified from the original sample. In gel electrophoresis, proteins and nucleic acids are separated. This process separates macromolecules based on their size and or shape and charge. Gel electrophoresis uses current flowing through a buffer to separate the macromolecules. So we have gel cassettes, this is how you would set it up. You would have liquid buffers and an anode and cathode. So positive anode, negative cathode. You would add your samples into the wells and then they would travel through. Native gels, so there's two different types, there's native and SDS page. Native gels separate the proteins by their size, shape, and charge. These native gels don't denature the proteins, so they keep their standard conformation and shape. This allows separation by shape, size, and charge. SDS page, on the other hand, separates proteins by size alone. Prior to SDS page will give all the molecules an equally negative charge and denature them. This will separate the proteins by size alone, as shape and charge have been removed. In a solution, a protein will change its charge based on the pH of its surrounding environment in the solution. This means that a solution's pH can be changed to find the point at which the protein has an equal number of positive and negative charges, so there is no overall net charge. A soluble protein precipitates from the solution at the point that is known as its isoelectric point, so it has no net charge. A mixture of proteins can be separated from a mixture using their different isoelectric points, their IEPs. If a solution is buffered to a specific pH, only those proteins which have an IEP of that pH will be precipitated out. So we can see this here, we've got 3, pH 4, 5, 6, 7, 8, 9, 10, and only those proteins that have the isoelectric point at that pH will precipitate. So soluble proteins can be separated using an electrical field and a pH gradient. They'll stop migrating through the gel at their IEP. This is known as isoelectrical focusing. So to summarize, centrifuges separate based on density, giving the pellet and the supernatant. Paper and thin layer chromatography can be used to separate different substances such as amino acids and sugars. More soluble solutes will travel further in a given time. Affinity chromatography uses molecules bound to a stationary column, which have a high affinity for some soluble molecule passing through the column. Gel electrophoresis separates charged molecules using an electrical field as they move through the gel. Native gels don't denature the molecules, so we separate by size, shape, and charge, whereas SDS page gives all molecules an equally negative charge and denatures them, separating proteins by size alone. Finally, at a given pH, only those proteins that have an IEP of that pH will precipitate out of the solution, as they have no net charge. This separation can be carried out during gel